Hello everyone. This is the another video lecture on sexual reproduction in flowering plants. And in the last video lecture, we studied about embryo, that is dicot embryo and monocot embryo, the development of the dicot embryo and then monocot embryo, the parts we studied. And embryo development, how the embryo development occurs, little bit I was explained about that, not that much because it is a quite lengthy process. And coming to the next, that is dicot embryo. Okay, here you can see the structure of dicot embryo. Okay, here this is the embryo genesis, the development of the embryo. This complete structure is the development of the embryo. Sorry, complete structure is the development of the embryo from here to here, you can see. And next to that, here you can see the structure of a dicot embryo. This is the dicot embryo. You can see the embryonal axis. Sorry, you can see the embryonal axis here, cotyledonal axis. Okay, in the upper region of the embryonal axis, or towards the upper region of the cotyledon, you can find the flumal or short tip, and this region we call it as the epicotyle. This already completed. And the lower region of the cotyledons, okay, towards the lower region of the cotyledons is a hypocotyle, and in the epicotyle region there is a presence of fluval, and in the hypocotyle region there is a presence of radicle and root cap. You can see here, this is the structure, important one. Just practice the diagram. And next to this monocot embryo, these are all the points regarding monocot embryo. And here the monocot, coming to the monocot embryo, here you can see the different parts. Here also the same axes are there. Usually here this is contains a single cotyledon, and that single cotyledon we call it as a scutellum. The single cotyledon we call it as a scutellum. And towards the scutellum, okay, there is an attachment of here you can see the coleoptile, and the shoot apex you can see here, that is a flumal. And along with that, on the other hand of the embryonal axis, we will find another cotyledon which is functionless, that is epiblast. Okay, and in the lower region of the embryonal axis, here you can see the radicle, root cap, and then coleorhiza. Coleorhiza is a covering. Okay, it's a covering of the root cap or root tip or radicle, and coleoptile is a covering of shoot apex or flumal. Okay, shoot apex or fluval, that we can see here. And this is about the structure of monocot and dicot embryo. And coming to the next aspect, students, that is seed. That is seed. Here we are studying about post-fertilization events. Post-fertilization events we are studying, and in that, first we studied about the endosperm formation. Sequentially we are studying sequentially. Next to that, embryo is going to be developed here. Next to that, seed development. Next to that, fruit development. This is a sequential events what we are studying. Okay, first one, endosperm development. That is very important because embryo completely depend on endosperm. Okay, embryo completely depend on endosperm for the nourishment. So first there is a development of endosperm, and next to that, here formation of embryo. Okay, how the dicot embryo and monocot embryo they are going to be developed. That is development of embryo. And next to that, here you can see the seed, the structure of seed and types of seed we can see here in this case. In this case, you can see the structure of seed and then types of seed. I will explain here the things about the seeds. Okay, here the seed. The seed we also call it as a fertilized ovule. Also call it as a fertilized ovule. We can say it as a fertilized ovule. Okay, here the seed to seed method occur here. Seed to seed. Okay, what is meant by seed to seed? What is meant by seed to seed? Here I am going to collect one seed. Here I am going to collect one seed. I am going to plant it, and it is going to be germinate, and there is a development of a plant. and this plant will get the maturity or reproductive stage, and it start producing the flowers, and fertilization occurs over there, and then finally they are going to be produce the seeds. Seed is nothing but it is the end product of a plant. It is the end product of a plant, that is seed. Finally there is a formation of seed. Okay, finally there is a formation of seed. How these seeds are produced? From the ovule. Okay, fruit gives rise to, sorry, ovary gives rise to fruit, and ovule gives rise to, sorry, ovary gives rise to, ovary gives rise to fruit, please remember, and ovule gives rise to seed. Ovule is giving rise to seed, and ovary gives rise to fruit. Okay, and the number of ovules can vary. Ovules are present inside the ovary. Ovules are present inside the ovary. The number of ovules can vary. For example, either it may be one, or it may be two, or it may be more than two, or it may be plenty, more than 100. You can see in case of watermelon. Okay, watermelon. The number of seeds present inside the fruit, that decides the number of ovules present inside the fruit. Okay, number of ovules present inside the ovary. How much ovules are present, that much seeds are going to be produced here. Okay, it means the number of seeds are dependent on the number of ovules. You can take the watermelon, you can find a plenty of seeds over there. It means it contains lot of ovules. Okay, it contains lot of ovules. And if you take the mango, only one seed is there. It means a single ovule is present over there. Or you can take papaya plant. In papaya plant you can find plenty of seeds. It means it contains plenty of ovules inside the ovary. Okay, it contains plenty of ovules inside the ovary. That is the concept that you have to remember. And here, coming to the structure, students, here they given the stage of last sexual reproduction in angiosperm. This is the last stage. Production of seed is the last stage of sexual reproduction angiosperms. Okay, it means the end product is the seed in case of angiosperms. And usually these seeds are developed inside the fruit. It means inside the ovary. Inside the fruit there is a development of seeds. Okay, or ovules. And if you observe the structure of the seed, it mainly contains a seed coat, it contains cotyledons, it contains embryonal axis. Already we studied about embryonal axis. Okay, first one here, seed coat. Seed coat is mainly made up of that integument. If you observe the ovule, the structure of ovule, you can see the structure of ovule like this. Okay, sorry, wait. Okay, like this. In the middle ovule is there. These are the integuments. These are nothing but integuments, and these integuments are the seed coats. These integuments are nothing but seed coats, and these seed coats are two in number here. Okay, two seed coats are there. If I draw the seed structure here, this is the seed, and the outer covering we call it as a testa. Outer covering we call it as a testa, and the inner covering we call it as a tegmen. Sorry, inner one we call it as a tegmen. Outer one testa and the inner one tegmen. We call it as a tegmen. And here the microfilar end is slightly open here. You can take the microfilar end here, because in the microfilar region these integuments are not covered. Okay, it is open here. The integument is the microfill is open here. You can see here, this is the opening of the microfill, and here there is a presence of testa and then tegmen in the seed. Okay, seed coats. This is about the seed coats. And if you see the structure, that is cotyledons. There is a mainly presence of either two cotyledons or single cotyledon. If they contain two cotyledons, those we call it as a dicotyledonous plants. If they contain only one cotyledon, they are called as monocotyledon. Based on the presence of number of cotyledons, we call it as a dicotyledon and monocotyledon. Okay, some plants contain two cotyledons, so those are comes under dicotyledons, and if some comes under, they some have only one cotyledon, so they comes under monocotyledon. Okay, this is the concept about the cotyledons. And the embryonal axis, already we know that embryonal axis, we can take the section, we can take consider one axis, that is the embryonal axis. We'll find the upper part, that is epicotyle, and the lower hypocotyle. So that already we discussed. This is about the embryonal axis. Okay, if you think about the endosperm in the seed, this is also already we studied. But if you see the seeds here, in some cases what happens, there is a albuminous seeds. Okay, there is a albuminous seeds. It means they persist endosperm. They have endosperm. The endosperm is not completely utilized by the embryo. Some sort or some bit of endosperm is remained in the seed. Such seeds we call it as albuminous seeds or endospermic seeds. Okay, albuminous seeds or endospermic seeds. As in case of wheat and then maize, we can take the example wheat and maize, and sunflower also we can take the example. Okay, because they contain endosperm in them. Okay, endosperm. Castor also you can take, castor plants also you can take, there is a presence of endosperm. Okay, such plants, such seeds we call it as albuminous seeds or endospermic seeds. And in some cases there is the absence of albuminous. Okay, it means the complete endosperm is consumed by the embryo. Okay, it is completely consumed by the embryo. Embryo is going to be consumed complete that endosperm. Okay, in such cases there is no endosperm present. Such endosperm we call it as a non-albuminous seeds. Such seeds we call it as a non-albuminous seeds or ex-albuminous seeds. Okay, or non-endospermic seeds. Example you can take here, pea and beans. We can take the example. Okay, this is about the albuminous and non-albuminous seeds or ex-albuminous or albuminous seeds, or we can say it as the endospermic and non-endospermic seeds. Okay, you can take the examples. Please remember the examples also. Okay, moving to next year, that is in some seeds, such as we can take the example here, black pepper and wheat. In case of black pepper and wheat, some amount of nucellus is going to be retained here, or it's remain present over there after the fertilization, after the development of the seed. Some sort of or some amount of nucellus is going to be remain here. That already we know the mass of cells present around the embryo sac, that we call it as the nucellus. Actually after fertilization and everything, okay, it is going to be disappear. But in some cases what happens, this nucellus remained, okay, persist after the fertilization and then to the development of the seed. It is going to be remain present over there. Okay, it remain present over there. And such seeds we call it as a perispermic seeds. Important, please remember the definition. What is perisperm? Perisperm is nothing but the remnant of nucellus. Okay, in seeds what happens, some amount of nucellus is going to be remain. Okay, remnants of nucellus, that we call it as a perisperm, or or such seeds we call it as a perispermic seeds. Okay, please remember about that. Okay, for your competitive exam also they will ask the question. Okay, that is known as perisperm. That is remnant of the nucellus is known as perisperm. Okay, and here somewhat here the integuments of the ovals are somewhat hardened here, that is a seed coat, and microfill facilitated the entry of oxygen in the seed. Okay, here you can see, I'll draw the seed diagram here. Okay, okay, here the seed present here. Okay, seed present here, the seed coat is somewhat hard because the external it is covered by testa and internally it is covered by tegma. Outer layer is testa and inner layer is tegma. And here you can see the microfillar opening. Okay, microfillar opening is present here. Through this microfillar opening, oxygen, water, okay, oxygen and water usually enters here. For the entry of the oxygen and for the entry of the water, this opening is very important, and this is mainly utilized by the seeds for the germination purpose. For the germination purpose it is mainly utilized here. That micropillar opening is necessary for the transport of oxygen and for the transport of water into the seed. For that purpose it is very important. And if you go through the dormancy, if you go through the dormancy, for example seeds, what will happen, they mainly lose the water. Okay, they will lose the lot of water from them. For example, if the seed fertilized embryo and the developed embryo, it mainly contains 75% of water. Okay, after complete maturation of the seed, only 10 to 15% of water remained here. Only 10 to 15% of water remained here. Okay, only 10 to 15% of water is remained here. Okay, remaining all the percentage of water is going to be uh, lost by the seeds. Okay, and they will undergo such a condition. Such a condition, what type of condition? They will undergo resting stage. They will undergo resting stage. They will lose the moisture and the seed may enter into the dormancy. Dormancy means here they are in resting stage. They are not active in this stage. Okay, in dormancy, dormant stage, we call it as a dormant stage. In this stage the seeds are not active. In this stage the seeds are not active. They are metabolically inactive. In this condition they are metabolically inactive because they require some favorable condition. Okay, because they require favorable condition. When they will got the favorable condition, they start to germinate. For example, temperature, for example moisture, for example oxygen. Okay, if they find such a favorable conditions for the development, and in that condition they are going to be start germination. But before that, what will happen? They will undergo dormancy or dormant stage. They are going to be undergo. It means in that stage the seed is inactive. It is inactive. There is no development or there is no germination of seeds. Okay, such condition we call it as a dormancy or a dormant stage we are going to be call it as. Okay, this is completely about the seed. These are all the points regarding the seeds. Okay, very simple concept is there, very simple methods are there. Okay, please remember those. And here they given the diagrams. Okay, I am going to show these are here. You can see here, this is the bee. This is the bean, you can say pea plant or bean, you can say here. There is no endosperm remained here. They are not labeled here any endosperm, only cotyledons they labeled here. Okay, there is only remaining of the cotyledons, there is no endosperm. But here you can say in the castor, this is a castor plant. This is the castor plant. Here you can see the endosperm. Okay, in the middle there is the presence of cotyledons, and in the surround, that those cotyledons are surrounded by they are surrounded by nutritive tissue, that is endosperm. This is in case of castor. And next one you can take here, that is onion. Here also there is a presence of endosperm. Everything, all cotyledon, seed coats are there, shoot apical meristems are there, hypocotyl is there, root cap is there, and along with that they also contain endosperm. Small amount of endosperm they have. And here this is the maize plant. Okay, this is the maize one, and here you can see the pericarp. That is the outer covering pericarp here, and here you can find the endosperm. Okay, here the endosperm present here, lot of endosperm you can observe in the middle. Same here, coleorhiza, coleoptile, coleoptile and coleorhiza you can find here, and here root cap and radical and then flumule, everything, every parts are present here. Along with that they also contain endosperm. Okay, this is about the complete information about seeds. Okay, very simple, and just remember the names. Okay, just remember the examples and names, and along with the definition of that what do you say, albuminous seeds and the non-albuminous seeds. Along with that you can remember the things about okay, that I will tell you. Perisperm. Okay, perisperm, please remember the definition of perisperm, it is also very important. And here, okay, this is the thing regarding seeds. I think all the points I have covered from your NCRT. Go through once uh, your textbook, that is NCRT textbook, and you will came to know about the concept. Very simple concept is there, that is seeds. Okay, and in the next video lecture we'll begin with fruits. Okay, which is a uh, which is a part of ovary, or which is developed from the ovary. Okay, that we'll study in the next video lecture. Okay, I think so all of you understood about this concept, and if you have any doubt you can ask, or you can comment in the comment section, or you can ask through the WhatsApp. Okay, thank you.